Welcome to EPG Pathshala. Today we will be learning about the general organization of nervous system and their neurons. After learning this module, you will be able to know about the general organization of the nervous system of the human body. Secondly, you would also know all the neurons present in the nervous system, their classification and their function. So, now we go on to the introduction. This module is basically is a very preliminary module where you will learn that how the nervous system of the brain is organized and what are the cells which are present, what are their types and what are their functions. So, what do we see in the general organization of the neurons and the nervous system? That the nervous system is a complex organ where it is there are divisions into the central nervous system and the peripheral nervous system. The central nervous system is the part which comprises the brain and the spinal cord, whereas the peripheral nervous system has two branches. One is the somatic branch and the other is the autonomic nervous system. The somatic branch is the one which innervates the limbs. The autonomic nervous system is the part which innervates the visceral organs. The somatic nervous system is further divided into two parts, where one is the receiving end, which is called as the effector, and the other is the part which sends the message, and it is the effector, and it is also called as the motor part. In the autonomic nervous system, which is in short is also called as the ANS, it has two branches, one the parasympathetic and the other the sympathetic. The parasympathetic is usually the one which takes care of our normal day-to-day functioning, whereas the sympathetic is the one which takes part in most of the emergency situations. So, that is the broad classification of the organization of the nervous system. Now, the whole nervous system is unique, which is made up of several kinds of cells, namely the neurons, the glial cells and the stem cells. Now, we can learn about them one by one. The neurons, which we see in the nervous system, are cells which are much larger than most of the cells in the body. They are complex structures which have a head and a tail portion. The head portion is the one which is called as the soma, whereas the tail portion is the one which is called as the axon. The axon ends in the terminal knobs. The soma or the head is like any other cell, it has a nucleus. Now, this nucleus is unique in that sense that it does not divide. The cells of the neurons remain in the quiescent stage for their lifetime till they die. And besides this, the soma also carries the other organelles in their cytoplasm. Now, these organelles are mitochondria which give the energy to the cell. There are endoplasmic reticulum, Golgi apparatus, ribosomes, which will participate in the protein synthesis as well as in the neurotransmitter synthesis, which is the prime function of these cells. Besides this, they also have a lot of cytoskeleton. In fact, cytoskeleton is the one which is responsible for the unique structure. The soma also gives out several projections, and these projections are named as dendrites or axons. Now, dendrites are different from axons because they are in structure, they taper, whereas the axons maintain the same dia throughout their length. The neurons are of several types, and which we will define in their classification. The axon, on the other hand, is the tail portion, and it is the one which is filled with exoplasm. Besides this, it has a lot of cytoskeletal elements, and these cytoskeletal elements help to transport the neurotransmitters which are formed in the soma, and these neurotransmitters are transported from the soma to the terminal ends from where they are released. The axon is also overlapped by, or there is a lining on these axons which makes them myelinated. If this wrapping is not there on the axon, then it is called an unmyelinated neuron, and these myelinated neurons work much faster than the unmyelinated neurons. And these myelin sheaths show a gap intermittently, and those are called as the nodes of Ranvier. And when the electrical activity crosses these myelin sheaths, they jump from one node of Ranvier to another node of Ranvier, and that makes a myelinated sheathed neuron faster acting than unmyelinated neuron. Besides this, the neuron also has to make connections with other neurons, and those connections are called as synapses. These synapses are connections between one neuron to another neuron, and they could be electrical in nature or they could be chemical in nature. Now, if we look at the classification of the neurons, there are different ways in which people have classified these neurons, and these neurons can either be classified based on their structure. Another method is to classify based on their function. Yet another method is to classify them based on the type of neurotransmitters which they release, or based on the electrophysiological function of the cell. Now, if we look at the structural classification, then it has been seen that there are 4 types of neurons. Like we call them the unipolar. The unipolar, those neurons which have one soma, one axon, and they have no dendrites. The other, the pseudo unipolar, which is actually a unipolar, but its axon is branched into 2. So, one acts as like a dendrite, the other acts like an axon. The bipolar neurons are similar to unipolar, but they have dendrites in their soma, and they are part of the, mostly they are part of the sensory system of the nervous system. Then there are the multipolar neurons, and these multipolar neurons have number of dendrites on their soma, and these are the usual neurons which are found in most of the nervous system. Now, if we look at the functional classification, then the neurons can be classified based on the their function, that either they receive the messages, then they are called as the effector or the sensory neurons, and they are usually the ones which receive the message. And then we have the motor neurons, which are the effector neurons, which carry the message from the brain to the effector organ. For example, it could be the muscle. And then there are interneurons, which connect the sensory neurons with the motor neurons. Those are interconnecting neurons. If we look at the reflex arc, then an arc consists of it shows an effector neuron and the effector neuron, that is the sensory neuron, the interneurons and the effector neuron. So, that is how they make a reflex arc. Now, another way of the classification is that the neurons, they release different neurotransmitters, and accordingly their functions are also different. We have several types of neurotransmitters, and the neurons which secrete are different. So, we can have several types. If it is a cholinergic neuron, it will release acetylcholine, and they are mostly excitatory in nature, and they are seen to be participating in cognitive functions. If we look at the GABAergic neurons, they are mostly they are releasing GABA, that is gamma aminobutyric acid, and these are mostly inhibitory in action, and they can induce, their action helps to induce sleep. Similarly, we have other type of neurotransmitters. Now, another classification which is based on the electrophysiology, in which we have different kinds of neurons, one which have which are tonically firing, or they are firing in a biphasic manner. So, we have different kinds of firing rates. Based on that, we have classified these neurons into 3 types. Now, as we go further in learning about the different kinds of cells in the nervous system, the next important neuron or the next important cell is the glial cell. Now, glial cells are the supporting cells of the nervous system. They do not carry messages, but they support the neurons, which are the prime effector cells. And in fact, 90% of the cells in the nervous system are the glial cells. They participate in giving nutrition to the neurons and to other glial cells, as well as to help in making the cerebrospinal fluid, to remove the toxin, to provide the immunity, and so on and so forth. They have number of functions. So, if we look at the different kinds of the glial cells, we see that there are 6 types. There are 4 types which are seen in the central nervous system, and 2 types which are seen in the peripheral nervous system. Like there are astrocytes, microglial cells, ependydymal cells, oligodendrocytes in the central nervous system, and there are satellite cells and the Schwann cells in the PNS. Now, they have different functions. For example, the astrocytes have been seen to be providing the blood-brain barrier. The microglial cells are participating in the immune cells. The ependydymal cells are important because they produce the cerebrospinal fluid. The oligodendrocytes, they form the myelin sheath of the central nervous system cells. Then we have the satellite cells and the Schwann cells in which of the PNS, where the Schwann cells provide the myelin sheath to the peripheral nervous system cells, whereas the satellite cells are also participating in covering the neuronal cells. So, what has been seen is that there are different kinds of cells, and it has been seen that there are number of techniques which can be used to study these neurons, and some of the neurons which have been studied has been studied by the techniques of histology, in which several kinds of stains which could be fluorescent stains which can also be used. Then single unit recordings where the electrophysiology of the neuron is recorded and the firing rates are also recorded, which helps the scientists to classify the neurons into different kinds. Besides this, electron microscopy has done wonders in providing lot of details of the structures which are present in the neuron. Now we see that the neurons and the glial cells are important cells, but as we have already said that these cells do not divide, and many cells they do die during the lifetime. Now the brain still has a system to renew itself, or some of the neurons which it loses, or if it wants to make new connections, there are cells called as stem cells present in the brain. Now those are called as the neuronal stem cells. Now these neuronal stem cells can form new neurons, they can also help to form the glial cells, and more above this, the neuron cells help to give plasticity in learning and memory. Now plasticity means adaptability, bringing new changes in the neuron so that it can participate in a particular function in a particular environment. So if we summarize this chapter, we find that the nervous system is mainly made up of the central nervous system and the peripheral nervous system. It is made up of neurons and the glial cells and the stem cells. And if we look at these classifications, their classification, we find in the structural classification there are 4 types: the unipolar, the pseudo unipolar, the bipolar and the multipolar. In the functional classification of neurons, we see there are sensory neurons, motor neurons and the interneurons. In the neurotransmitter secreting neurons, we have several types. There are as cholinergic, GABAergic, glutamatergic, serotonergic and several types. If we look at the electrophysiological classification, we find that they can be the fast firing type, they can be the tonic types, and most of these studies have been done using the staining techniques or the recording solid single unit recording, or the electron microscopy has been used to study these neurons. Thank you.